my name is Andreas Hougaard Laustsen-Kiel. Very horrible name, even in my own language. I'll be talking about snakebite envenoming and antivenom today. To start out with, I'd like just to show that this is of course not work I do alone. I run a lab at the Technical University of Denmark. So it's a lot of students, postdocs, and researchers and lab technicians that are behind all of what we do. But I'm a professor in antibody discovery, and that gives me a special set of skills that allows me in a picture like this, where for the untrained eye, it can be very difficult to see where an antibody is, to write out in the wild and quickly see where is the antibody here. So you could say that's my area of expertise. And more interestingly is what do I then use this skill set for? And as today's talk title will reveal, it'll be a lot about toxins today and antibody, and in particular antibody cocktails. Snake venoms are very complex mixtures of toxins, and we'll get a little bit into sort of how do they work and what do they do, and then what can we sort of do about it. But before jumping into, you could say, the technicalities today, I'd like to take you back to 2011, where I was on safari in Tanzania. I was not there in particular to see snakes or anything like that, but just wildlife. But on this trip, I came across a snakebite clinic where two victims that were approximately 5 and 8 years old had been bitten by spitting cobras. And despite reaching this clinic, they had no other sort of option than amputation available. So one kid had been amputated around the knee and the other one at the elbow. But besides that, which is of course serious enough in itself, we're lucky to survive a otherwise lethal envenoming. So this obviously made a very big impression on me and ended up having a big impact on my later scientific career. So there's going to be a few quite nasty pictures just so that the people are aware. But to give you an example of what I sort of saw, this is not the particular kid, but it's a similar type of envenoming by a spitting cobra, is that toxins from this particular venom, when they are injected into the body of a victim, will cause massive tissue destruction, necrosis, and really bad scarring, which in this case, it may look bad to many of you, but it's not even particularly bad. So that's one type of action that snake venoms can have, is sort of tissue destruction and necrosis. Another classical sign of snakebite is internal hemorrhage, massive bleeding. In this particular case, viper toxins go in and they interact or interfere with the blood's ability to coagulate, which causes clinical manifestations like the one you see in this picture. And the last classical hallmark of snakebite envenoming is paralysis. In this case, a child has been bitten by another type of cobra, which causes neurotoxic effects, which manifest themselves in paralysis. And in the worst cases, it leads to an inability for the victim to breathe because the muscles around the lungs are sort of inhibited in their function. To give you an example of what happens on the sort of molecular level, in case of neurotoxicity, toxins from some venoms, they go in, they interact with ion channels, they inhibit their function, and this completely blocks what is called neuromuscular transmission, or basically our ability to move, including breathing. But if we zoom out and don't just look at the individual snakebite, because I think probably for most people here today, it's clear that getting bitten by a snake is a bad idea and it can be very serious. But if we zoom out and look at it from a global perspective, it's not just a problem for the individual. It's actually a problem for many societies, particularly rural societies in the global south, where people, they are dependent on manual labor to sort of bring home the bread, so to say. And here, people are in close contact with many very venomous snakes, either when they're working or when their children are playing in the field. And this means that several million people each year are envenomed by snakes, which leads to around 100 100,000 deaths and about 3 to 4 times as many amputations or other types of permanent handicaps. But perhaps let's go back to talk about what is actually snake venom. And I think a very important division is between venoms and poisons. In the English language, we have such a distinction. Most other European languages, we don't. We just all call it something like gift or similar words to that. But whereas venoms, they are protein-based and you could say toxic mixtures that need to be injected either by fangs or some kind of stinger device. Poisons are typically small molecule based and are toxic both when injected but also when ingested. And that means that poisons are what we find in poisonous fish and frogs and many plants and mushrooms. And because they're small molecule based, that means that when you ingest them, they can be taken up over the intestinal barrier and cause toxicity. But venoms are protein based and they need to be injected simply because they are not stable in the gastrointestinal tract and they cannot easily enter, you could say, the human body across the intestinal line. So this has the implication, because they're protein-based, that we can use them for immunization. And this is exactly what is done in in antivenom production. Antivenoms have existed for 120 years. And if we go through how we make antivenom, we start by obtaining a snake. Typically, it's obtained originally from the wild, but from there on kept in captivity. Then it's milked for its venom. The venom is used for immunizing a larger animal, typically a horse or a sheep. Once this larger animal has built up immunity, so that means a lot of antibodies in its blood that can recognize the toxins present in the venom, we draw the blood, we fractionate the blood into its different components, we remove the red blood cells, isolate the antibodies present, concentrate them a bit, and then formulate them into an antivenom or sort of a small vial. And isn't this just perfect? Well, if it was, I would be out of a job. At least a large part of my lab would have nothing to do. So some of the problems with this is when you make antivenom this way, despite that it is effective at neutralizing venom, a large part of the antibodies present, they don't actually recognize the snake toxins. The horse or the sheep will have encountered all sorts of other things throughout its life. And that means that many of the antibodies are totally irrelevant. Are totally irrelevant in to treat an envenoming. Moreover, all these sort of antibodies, they are typically horse or sheep derived. And when you take something from an animal and inject it into a human recipient, the human immune system will recognize it as foreign and may launch sort of a counterattack simply because it recognizes that this is not what we usually have in our body. So despite that antivenoms, they can save lives and they are effective at neutralizing venom, they come with these different drawbacks, and this makes them not as efficacious as they should be. And it also means that they come with certain side effects, such as serum sickness, or in the worst cases, anaphylactic shock, which can be life-threatening in itself. And to put it into perspective, if you're bitten by a large black mamba, Africa's largest venomous snake, you will need almost 50 grams of dry weight antibodies from a horse. And in comparison, if you have late stage colorectal cancer, the amount of antibodies you will receive in some of the sort of more modern treatments is less than a gram of human antibodies. So with antivenom, it's not so much of a question of whether or not you get side effects, it's how bad they are and will they potentially kill you. So antivenom can only be administered at hospitals where you can deal with things like allergy and severe anaphylactic shock. So while traditional antivenom is made in the way I just described, what we try to do in my lab is to essentially do the same as what was done with blood factors and insulin and certain other sort of therapeutic proteins, of moving away from the original natural source, in this case, horses and sheep, and and go and use some of the different techniques and methods we have in biotechnology and make human antibodies using recombinant DNA technology. So finding genes for specific human antibodies that can neutralize snake toxins and making them using cell cultivation, fermentation, and essentially replacing the horse antibodies with human versions. So to do this, I originally had to go to Costa Rica. I had to sacrifice myself in the name of science. I went to the labs of Professor Gutierrez and Lamonte, who are experts in analyzing snake venoms. And this is a view from my office. It was extremely tough, I can promise you. But here I brought the venom from a very notorious snake, the black mamba that I mentioned before. A snake that can reach more than 4 meters. It can raise its body 50% off the ground and look down at a short guy like myself. It's known for having a super potent, rapid-acting neurotoxic venom, always injecting venom in its bite, biting multiple times and being extraordinarily defensive. So not a snake you would want to sort of disturb in the wild. But without going into too much detail, we separated the venom into its different components. And what you see in this picture here is a so-called chromatogram, where each of these different peaks that have gone through an advanced filter system contains a number of different toxic proteins. And I've highlighted that some of these are very potent against mammals, so that includes humans, whereas fortunately some of the other peaks that we see are not so effective against mammals, but might be more effective against avian prey or amphibian prey or other things. So a big part of my work in Costa Rica was to find which toxins is it really we need to neutralize in the case of a human envenoming. Wasn't completely without trouble, but we managed to solve the different troubles that we encountered along the way. And that allowed me to then instead go a little bit closer to my base in Denmark. This time I went to Cambridge in England, where I had the pleasure of working with Dr. McCafferty and Dr. Anish Karat-Balat, who are experts in antibody discovery and really taught me how to use the technique called Phage display. As you'll see, Cambridge is a little bit different than Costa Rica, so this was more how it looked there, just for those of you who are interested. As said, McCafferty and Karat-Balat, they taught me how to use a technique called Phage display that received the Nobel Prize in 2018. And without also going into too many details here, it's an in vitro technique where you can essentially simulate the human immune system in the laboratory. And what we do is we connect snake toxins to the bottom of a well, and then we subject these toxins to a library of antibody displaying bacteriophages. And we have extremely large libraries with several billion different antibodies. And when we're lucky, there'll be some of these antibodies that stick to the toxins. Those that don't can be washed away. We can amplify these different phages and do another round to really find the best binders. And because these phages contain the DNA that encodes the antibody, we can sequence and figure out what antibodies actually bind to these different snake toxins. And using this technique, we were able to design 2 different cocktails, one that contained 4 different antibodies and one that contained only 3, that were able to neutralize all these different toxins that were present in the venom of the black mamba. And what we show here on the right-hand side, which is probably the most exciting of the graphs, is that these 2 different cocktails, depending on the dose, were able to protect mice that were challenged with the entire venom from the black mamba. They were able to completely protect these mice so that they could keep on living. And that was, I think, in many ways, my big scientific breakthrough, showing for the first time that human monoclonal antibodies could actually be made completely in the lab to neutralize snake toxins and let alone an entire venom. And I think with that, I just want to sort of show that many different collaborations and people went into this work that I'd like to thank. Definitely not a one-man job. And I would like to say thanks for your attention and then open up for questions if there's time for that. So thank you.
Thank you very much, Andreas. There certainly is time for questions. There's time, about 5 minutes for questions. So is there, people can put their hand up or just unmute and ask something. Yes, Valeria, you've got a question.
Hey, very nice. I didn't know much of it actually. And I was curious, how much time do you actually have to inject the antivenom to be effective? And also another small question, because you mentioned that you basically saw the success in mice, and you generated the antibodies from human tissue, right? And so it's interesting that they worked. So don't you expect didn't you expect some strong reaction from the mice, like it would happen if you inject a horse into the humans? Yeah, sorry, it's a bit naive,
yeah, but super good questions. To take the first one in terms of timing, it's a question I often get, and I think the most important thing to realize is a snake is not a trained physician. It doesn't deliver the same dose. It delivers it different anatomical parts, and different snakes have different types of toxins and different venoms. So it's always hard to estimate exactly how quick will you die, or when will symptoms develop. So I would say, if you're very unlucky and you're bitten directly in a vein, which happened unfortunately to a plantation worker while I was in Costa Rica, then you might die within 15 minutes. But that is a very unlikely case. Normally you inject it in muscle or fat tissue, and it can take a number of hours before the first symptoms develop. It can sometimes take almost 24 hours before the clinical manifestations really appear. And what you say is, I mean, of course, it's best to get antivenom as fast as possible, but antivenom can help in up to 48, maybe even 72 hours for some snake bites. A black mamba is the one I work with. You're probably on the lower scale of that it's better to get it within a handful of hours, because it is a potent and rapid-acting neurotoxic venom. But you do have enough time for most bites to treat them if you are within hours of a hospital. That being said, time is of essence, of course, and you have a golden window. The other question of whether the mice would react to the antibodies. You're right, of course, there could be an issue with injecting human antibodies, and that the mice would get also anaphylactic shock to them. Without going into too many details, the antibodies from humans don't seem to cause as much trouble in mice. And that's a little bit of an evolutionary weird thing, because mice antibodies do it in humans. So it's a one-way street, that the human so-called FC part of antibodies is compatible more or less with mice, but not vice versa. Also, especially in the type of assays we do here, where you look at neurotoxicity and lethality, you would be unlucky to see severe anaphylactic shock in mice. But that being said, it's a good question. It might happen. Yeah. We haven't observed it I think in any of the experiments we've done. Yeah, I hope, does this answer your question?
Yeah, sure. I was just curious.
Yeah.
Thank you, Valeria. Arild.
Yes, thanks, Andreas, for a nice talk. So if I understand right now, then essentially what happened was that you would get bitten by a snake, then you have to inject the antivenom. But is there any possibility for sort of long-term sustained immune responses so that you essentially could get a vaccine for, being protected against being bitten by snakes?
You could say technically it could be done, but it would be the world's most complex vaccine. A typical snake venom has something like 50 different toxins, and maybe it's not all the toxins you need to have protective antibodies against, but you definitely typically need against a handful or two. Snakes have very different venoms. There are very few toxins that you find across different species. Do you know which snake you're going to be bitten by, or do you want to have a very advanced vaccine with 100 different components? If you look at the horses, many snake toxins are not very immunogenic, so you need repeated injections every third week or so for more than a year. So it would be a complicated both vaccine to design, but also a complicated vaccine to use. And even in the most snake-infested areas, it's only 1 out of 2,000 people who are bitten by a snake every year, or maybe it's die of a snake. But that's of course a high number when you look at deaths. But it still means that you would need to vaccinate at least 2,000 people, and in most parts of the world, it would probably be 100,000 people per snakebite. So even if a vaccine would be cheap, which I doubt it would be in this case, but if it was, you would have to multiply the cost with quite a lot to protect everyone, because it's of course hard to predict exactly who would be bitten. And I think it's of course also relevant to mention that, stating the obvious, snake bites, they are not infectious. So there's no herd immunity benefit, as we know from other vaccines against infectious diseases. So it would be a personal vaccine that only helps the victim.